Our next speaker is Jyoti Iyer from the NIH, who's going to tell us about PP1b, controlling ZYG-1 levels to ensure precise centrosome doubling.
Hi everyone, I'm very grateful to the organizers for giving me the opportunity to present my work in front of all of you. So my name is Jyoti Iyer and I'm a postdoc in Dr. Kevin O'Connell's lab at NIDDK. Today I'm going to talk to you about how protein phosphatase 1 ensures precise centrosome doubling by regulating ZYG-1 protein levels. So let me start out by telling you what centrioles are. These are microtubule-based organelles, and their number is tightly regulated. So in the cell, there are usually 1 to 2 pairs of centrioles that are present. Each centrosome, which as you know is a microtubule organizing center of the cell, is comprised of a pair of barrel-shaped centrioles, including a mother centriole, which is the older of the 2 centrioles, and a daughter centriole. And these are surrounded by an amorphous electron-dense material called pericentriolar material, or PCM. So it's very important that centriole duplication occurs accurately, such that one and only one daughter centriole is formed adjacent to each mother centriole. And deregulation of this process can lead to abnormal cell division and cause aneuploidy, which is a hallmark of cancer cells. So the centrosomes organize the mitotic spindle, and in quiescent cells, they also move to the cell surface where they serve as basal bodies for cilia assembly. Now it's very important that centriole duplication occurs once and only once during each cell cycle. So during S phase, along with the chromosomes, the 2 centrioles also duplicate, such that at G2, you have 2 duplicated sets of centrioles and 2 sets of chromosomes. During the metaphase stage of mitosis, the 2 centrosomes move apart and establish poles of the mitotic spindle, which ensure precise chromosome segregation upon cell division. At anaphase, the mother and the daughter centriole disengage, and they are licensed to undergo subsequent rounds of duplication. Now, several diseases are associated with defects in centriolar and ciliary genes, including MCPH. So it's very important to understand how the centrosomal proteins are regulated. Now, in our lab, we use C. elegans as a model system to study centriole duplication. So in C. elegans, the sperm brings the first centriole pair to the egg, so that upon fertilization, these sperm-derived centrioles separate, they duplicate, and they continue to establish bipolar spindles at each cell division during embryogenesis. However, whenever there is a maternal loss of any of the core centriole duplication proteins, what happens is that these sperm-derived centrioles are still able to separate and establish a bipolar spindle at the 1 cell stage, but because they fail to duplicate at the 1 cell stage, at the 2 cell stage you see monopolar spindles. So whenever you see monopolar spindles at the 2 cell stage, it is indicative of a failure of centriole duplication, and this will be important to remember for the rest of the talk. So now the components of the duplication pathway were first identified in C. elegans, and all the proteins that have been shown to be required for duplication in C. elegans thus far have corresponding homologs in other organisms, including humans and flies. So in C. elegans, 5 proteins have been shown to be required for centriole duplication. Of these, it's important to remember that the kinase ZYG-1 is a functional ortholog of PLK4, and it is a master regulator of centriole duplication. So in our lab, we isolated a mutation in the ZYG-1 gene called ZYG-1-IT25. This is a hypomorphic temperature-sensitive point mutation in the ZYG-1 gene, which greatly reduces its activity at the restrictive temperature. So in these animals at the restrictive temperature, duplication fails, and this yields monopolar spindles at the 2 cell stage. So I'm going to play movies for you now in which the DNA is labeled red with M-cherry histone, and GFP-SPUT2 is a centrosomal marker. So in wild-type embryos at the restrictive temperature, duplication proceeds normally, and therefore upon cell division at the 2 cell stage you see that each cell has 2 centrosomes. So this is what one would normally expect. However, in the ZYG-1-IT25 mutants at the restrictive temperature, duplication fails, and this causes monopolar spindle formation at the 2-cell stage. So ZYG-1 activity is very important to ensure that duplication occurs normally. And because of this failure of centriole duplication in the ZYG-1 mutants at the restrictive temperature, these worms produce 100% dead embryos. So what we did is, in our lab, we performed a suppressor screen for the ZYG-1 IT25 mutants using chemical mutagenesis. And then we tried to identify extragenic suppressors which now restored viability at the restrictive temperature. So we refer to these genes as SUSY genes for suppressor of ZYG-1. So using this screen, we identified two genes, SUSY2 and SUSY6. And interestingly, both these genes encode conserved regulators of protein phosphatase 1. So what is SUSY2? This gene encodes for the conserved PP1 regulator inhibitor 2 or I2. So I2 is an intrinsically disordered protein, and what it does, it binds PP1 at 3 different sites, and it blocks the binding of substrate to the catalytic site of PP1, and further binding of I2 to PP1 also displaces the metal ion from the catalytic core of PP1, thereby further inactivating it. So I2 is a well-known inhibitor of PP1. However, it is also known that phosphorylation of I2 on a particular threonine residue causes it to switch from being an inhibitor of PP1 to being an activator of PP1. So I2 can either inhibit or activate PP1. So we wanted to know, in C. elegans, does I2 function as an activator of PP1 or it functions as an inhibitor of PP1? So to answer this question, we isolated a mutation in the I2 gene called SUSY2BS4. This is a G2A splice donor site mutation which reduces I2 protein levels. So we looked at the phosphorylation of histone H3 at serine 10 in both wild-type embryos and in the I2 mutant embryos. So I must mention that phosphohistone H3 serine 10 is a well-known substrate for PP1. So the greater the phosphorylation of histone H3 at serine 10, the lower the activity of PP1. So what we saw was, in the I2 mutants that have low levels of I2, PP1 activity is greatly reduced. And this suggests that normally, at least in C. elegans, I2 predominantly functions as an activator of PP1. And we were also able to verify this by Western blot analysis. Next, we wanted to look at what effect this mutation in I2 has on centriole duplication. So we monitored centriole duplication either in the ZYG-1 IT25 mutants alone or in the ZYG-1 mutants carrying the I2 mutation, and both of these were looked at the restrictive temperature. So again, the DNA is red in color and the centrosomes are green in color. So you can clearly see that in the I2 mutants, in the ZYG-1 IT25 mutants, as expected, at the restrictive temperature, duplication fails, and this causes monopolar spindle formation at the 2-cell stage. So this is what we would expect to see. However, what was interesting was, when we introduced the I2 mutation in this background, this was now able to restore proper centriole duplication in the ZYG-1 mutants. So we went ahead and quantified this, and again we saw duplication fails in the ZYG-1 mutants alone. However, if we reduce I2 levels, either by introducing the BS4 point mutation or by performing RNA-I for I2, in both these cases, we were able to partially restore proper centriole duplication in the ZYG-1 mutants. So this suggests that I2 functions as an inhibitor of centriole duplication. So the other gene that we identified in our suppressor screen was SUSY6. This encodes another conserved PP1 regulator called SDS22, which is a leucine-rich repeat domain-containing protein, and it is highly conserved. So SDS22 is known to physically interact with PP1 and is required for proper chromosome segregation during mitosis. And importantly, like I2, SDS22 has also been shown to be required for complete activation of PP1 in vivo. So we isolated a mutation in the SDS22 gene called BS9. This is a G224-S point mutation in the conserved leucine-rich repeat region of the SDS22 protein. So we wanted to know what effect this mutation in SDS22 has on centriole duplication again. So we monitored centrosome duplication either in the ZYG-1 IT25 mutants alone or in the ZYG-1 SDS22 double mutants, or in ZYG-1 mutants in which SDS22 had been knocked down using RNAi. So as you can see, again as expected, at the restrictive temperature, duplication fails in the ZYG-1 mutants. However, whenever we impair SDS22 function or we decrease SDS22 levels using RNAi, in both these cases, we were able to again partially restore proper centriole duplication in the ZYG-1 mutants. So this suggests that, like I2, SDS22 also functions as an inhibitor of centrosome duplication. So next we wanted to verify whether we could detect an interaction between PP1, I2 and SDS22 in C. elegans. So we performed immunoprecipitation assays to test this, and you can see here that the PP1 pull down was able to pull down I2 as well as SDS22. So PP1 interacts with both I2 and SDS22. Also a reciprocal IP of I2 was able to pull down PP1. But interestingly, what we saw was that the I2 IP did not pull down SDS22. So this suggests that possibly both I2 and SDS22 form separate complexes to regulate PP1 activity. So since we had identified I2 and SDS22 in our suppressor screen, the next logical question that we wanted to ask was, does PP1 itself, the loss of PP1 itself also suppress the centriole duplication defects of the ZYG-1 mutants? So to answer this question, we looked at the effect of PP1 isoforms GSP1 and GSP2 on centriole duplication. So in C. elegans, there are two PP1 isoforms that are expressed in the worm embryos. GSP1 is a homologue of human PP1beta, whereas GSP2 is a homologue of PP1 alpha. So what we did was we first knocked down GSP1 using RNAi with about 90% efficiency, and this knockdown did not have any effect on the levels of the other PP1 isoform GSP2. Then we again monitored centrosome duplication. Again, as you expected, at the restrictive temperature, the ZYG-1 mutants failed to duplicate their centrosomes. However, when we knocked down GSP1 using RNAi, we were able to partially restore proper centriole duplication in the ZYG-1 mutants, just like what we saw with I2 and SDS22. And surprisingly, the introduction of a putative GSP2 null allele in the ZYG-1 background did not have any effect on centrosome duplication. So this suggests that GSP1 and not GSP2 is the isoform of PP1 that is important in regulating centrosome duplication in C. elegans. So like I2 and SDS22, we found that GSP1 also inhibits centrosome duplication. So next we wanted to know what is the biological relevance of this regulation of centrosome duplication by PP1. So to answer this question, we looked at embryos from SDS22 transheads in which GSP1 activity was greatly reduced, and these are 100% embryonic lethal. So in these movies, again, the DNA is red and centrosomes are green in color. So what we saw was that until the 2-cell stage, we did not see any defects in centrosome duplication. However, starting from the 4-cell stage, we saw a robust overduplication of centrosomes. So this was really exciting to us because this is the first report of a robust and consistent centrosome overduplication phenotype in the C. elegans embryo. So we decided to take a closer look at this phenotype, and we went ahead and quantified the duplication errors. What we found was that whether we decreased GSP1 activity by either constructing SDS22 transheads or by making a mutation in I2, no matter which route we used, we saw a consistent overduplication of centrosomes starting from the 4-cell stage. So in the SDS22 transhead embryos, about 40% of the centrosomes overduplicated starting from the 4-cell stage. And we see the same phenotype in the I2 mutants, although this is a bit weaker than the SDS22 transheads. So if the error were to occur during the first round of centriole duplication, we would have expected to see overduplication of centrosomes starting from the 2-cell stage. But as you can see from this data here, we did not see any defects in centrosome duplication at the 2-cell stage. So we conclude that the error in centriole duplication is happening starting from in the second round of centriole duplication, which results in centrosome amplification starting from the 4-cell stage. So we do not know why this is the case, but we felt that this is an interesting observation. Next, we wanted to closely look at the centrosomes from the SDS22 mutant embryos. So we performed structured illumination microscopic analysis of these embryos. And what we saw was that in some instances, we saw normal-looking centrosomes in which, in this case, a single daughter centriole assembled next to a mother centriole. However, there were also instances in which we saw multiple daughter centrioles assembling next to a single mother centriole. So in this case, we saw 2 daughter centrioles assembling adjacent to a single mother centriole. So again, this was really exciting to us because it told us that GSP1 activity is very important to make sure that one and only one daughter centriole forms adjacent to each mother centriole. So when GSP1 activity is reduced, the centrioles overduplicate. So far we know that SDS22 and I2 both activate GSP1, and we know that GSP1 inhibits centriole duplication. However, at this point we do not know the mechanism by which this happens. So the answer to this question came from previous studies that were carried out in both human cells, Drosophila, and some other systems. So in this particular study, they showed that overexpression of the functional ortholog of human ZYG-1 called functional ortholog of ZYG-1 called PLK4 in humans caused appearance of centriole-like structures in human cells, in U2OS cells. So this was very similar to the phenotype that we were seeing upon lowering GSP1 activity. So we suspected that possibly lowering GSP1 activity is having an effect on the levels of ZYG1. So we tested this by performing Western blot analysis for ZYG1 in wild-type embryos and in the I2 mutant embryos, which again have low levels of ZYG1 activity. Low activity of ZYG1. And as you can clearly see, we saw an overexpression of ZYG1 in the I2 mutants. In fact, we saw as much as a 4-fold increase in ZYG1 levels in the I2 mutants. So this suggests that GSP1 functions to downregulate ZYG1 levels. So in the I2 mutants, when GSP1 activity is reduced, ZYG1 levels go up. So what we feel is happening is that when GSP1 activity is reduced, ZYG1 levels are elevated, and this causes the overduplication of centrioles. So we wanted to know at what level this regulation of ZYG1 protein was occurring at. So we looked at both transcription of ZYG1 by performing QRT-PCR in wild-type and I2 mutants, and we did not see any significant difference in ZYG1 transcript levels between the two. Therefore, we concluded that GSP1 does not regulate ZYG1 transcription or message stability. We also looked at translation of the ZYG1 protein by constructing a GFP histone fusion that was driven by the ZYG1 promoter and under the control of the ZYG1 3' UTR. Other labs have shown that this can be used, this is a method that can be used to reliably predict the translation of a protein through the 3' UTR in the C. elegans germline. So if GSP1 were regulating ZYG1 translation, we would expect to see an increase in fluorescence intensity of GFP-H2B in the I2 mutants, but we do not see that. So this suggests that GSP1 does not regulate ZYG1 translation. So these data collectively point out that possibly GSP1 regulates ZYG1 levels post-translationally. So our current model is that SDS22 and I2 both activate GSP1. GSP1 then either directly through physically binding ZYG1 or indirectly through some other protein causes downregulation of ZYG1, and this inhibits centriole duplication. So in summary, today I have shown you that GSP1, I2 and SDS22 all negatively regulate centriole duplication. GSP1 downregulates ZYG1 levels, and this PP1beta or GSP1 mediated pathway functions to ensure that one and only one daughter centriole forms adjacent to each mother centriole. The regulation of ZYG1 levels by GSP1 seems to be post-translational. So in the future, we would definitely like to identify the substrate of GSP1. I did not have time to show you this data today, but we have performed a series of IP mass spec experiments for both GSP1, ZYG-1, and also the GSP1 regulators. We could not detect an interaction between GSP1 and ZYG-1 using those assays, but this could also be because the interaction could be transient. So currently we are in the process of utilizing a more sensitive biotin ligase-based interaction assay to determine whether we can detect an interaction between PP1 and ZYG-1. We would also like to determine the mechanism by which dephosphorylation of the GSP1 substrate causes degradation of ZYG-1. And finally, we would like to identify the kinase which is involved in phosphorylating the GSP1 substrates that causes ZYG-1 downregulation. So with this, I would like to thank my supervisor, Dr. Kevin O'Connell, for all his guidance and support throughout this project. I would like to thank Nina Peel, who is also, she has contributed equally to this study. I would like to thank our technician, Yan Liu, members of the O'Connell Lab, members of the Baltimore Worm Club, and our NIH colleagues for critical feedback and comments regarding our work. Finally, I would like to thank CGC and the Japanese Knockout Consortium for strains. And with this, I'll take any questions that you might have.
Maybe I can start. Can you speculate why this effect occurs like after the second cell division, like at the 4-cell stage? Do you have any explanation for that?
So we don't have enough data for this, but we have an idea of why it might be happening. So we have mutants of ZYG1 in which ZYG1 is active in the sperm. And so when we cross these mutants to our, the SDS22 transheads, we now see overduplication starting from at the 2-cell stage. So we feel that there is a threshold level of ZYG1 that needs to be reached in order for us to see overduplication. So when we activate ZYG1 earlier, we see overduplication earlier. So, but we don't have enough data on this, so I can't be extremely certain, but we feel this is what is happening.
Okay, if there's no more questions. Thank you.